the invitation to come speak. In today's webinar, I want to tell you about HIRN, but before I do, I'm not sure how many people in the audience actually know about type 1 diabetes, so very briefly, I want to give you a background. The picture you see there is a cartoonized photo of a human pancreas. And this is probably one of the most complex organs in the human body, but it's also very vulnerable. It can be easily damaged and attacked. Now, inside the pancreas, there are these collections of cells known as an islet. And what islets do are they regulate your blood sugar level. And in particular, when we look at those green cells called beta cells, the green cells help secrete insulin, which is a major hormone responsible for lowering your blood sugar. Now, normally, beta cells make lots of proteins, and those proteins get taken up by the vacuum cleaner, and that's called a dendritic cell. And that's usually the end of the story. But sometimes what happens are that these proteins start being shuffled over and taken to other immune cells. And what these immune cells do are one of two things. They either start communicating with B cells and say, hey, start making antibodies against these beta cells, or they start talking to these cytotoxic T lymphocytes and say, hey, kill the beta cells. And so that's obviously not what we want. And so what we can try to imagine is if we could figure out how to manipulate other immune cells to suppress this problem, then we could avoid a major disaster. And so what I want to tell you about is the kind of research being done to address type 1 diabetes. And this is probably the most scientific I'll get today. So back in 2014, a very large network was set up with many different focal groups. And this was a very large investment and still is by the National Institutes of Health. And so what I'm going to do is walk you through these different focal groups or consortia in HIRN. And the first is called CHIB, the Consortium of Human Islet Biomimetics. And that's simply taking a human islet and trying to put it on a microchip, and that would enable us to expose it to different drugs, different reagents, and try to monitor and measure what the reaction is. Think about personalized medicine. Another group is looking at how you can target a beta cell and get those beta cells to regenerate. A third group is looking at actually how you can keep these beta cells alive and prevent them from dying. There is one group that we'll talk about today in a little more detail that's really focused on characterizing the pancreas in a very deep way, deep phenotyping of the pancreas. A major focus is trying to understand how these beta cells interact with the immune system, as in the slide that I had just presented to you. And of course, our role, as Kawei had mentioned, is to coordinate the data and coordinate the administrative aspects of this group. So now you know there are 6 different groups, and what have we learned? Well, as I said, we're not going to talk about the science today. It's not the focus of the presentation. But if you wanted to read about the progress, you can go online to our website, and there have been some executive summary reports prepared to simplify the science. But if you wanted to get into the detail of the science, there's actually 400 plus publications there as well. So there's plenty of progress being made and lots of exciting new developments. But today's focus is about getting your hands on the data and what you can do to do that. So what we're going to do is we're going to walk through a few different data portals and show you some usage examples of how you can use these different resources. Now, the first is called the HIRN resource browser. We have a little over 1,000 different resources, and we'll describe those to you in just a minute. The second is this pancreas database, and this has close to 30,000 different files, ATAC-seq, RNA-seq, imaging mass cytometry, lots of different variety in terms of the data related to characterizing the pancreas. The third portal I'm going to tell you about today is this Pancreas Atlas. And this is focused on taking the imaging data and making it more accessible and analyzable to you to explore that further. And the fourth focuses on an aspect of the earlier slide I showed you where you're looking at these different regulatory cells or other types of immune cells and looking at the identity of these cells and if you can find anything unique about those. And I'll describe that in a little bit. So first, let's go to the resource browser. As I described to you, there were 5 scientific consortia in HIRN. Each are utilizing diverse resources and generate multidimensional data. And one of the things we did early on was begin to catalog all of these resources. And so you can go online to our website, and if you just want point and click access, in other words, you're not programmatically savvy, you just want to use your mouse to access everything, you can do that. So here are some questions that you can ask. Maybe you're looking for a polyclonal insulin antibody. You can search that here, see what we have, click on it, identify a particular resource. But then you might ask, well, how was that antibody used? And so you can see what the application or applications of that particular antibody are. In this case, it was an immunostaining assay. But you might also wonder, well, what other reagents were used along with this particular antibody? And so what we've done is we've also detailed maybe other antibodies that were used in the experiments here, or maybe there were different organisms that were used, whatever were related to that resource. From an experimental point of view, we've also linked that up as well. But you might say, no, this isn't good. This isn't what I want. I need a monoclonal antibody instead. And you might go and search the monoclonal antibody and say, oops, well, HIRN doesn't have one. Well, in that case, what we've done is we've sent you over to dkNET to look for that resource there, because as probably some of you on the phone know, dkNET has a very large collection of resources, and what we're focused on is just the resources within HIRN. But usually resources aren't ready just to be cataloged unless they're first curated. And so really briefly, I want to show you how we do that. This is a process by which we take bioreagents, so just one slice of the data that we are focused on, and then taking that and registering that data and putting that data into an authoritative database. Now, that middle section that you're looking at is very familiar to dkNET. They're the experts, and they've been doing this for quite a while. And so how do we take advantage of that? Well, early on, we started working with dkNET to start to identify some key metadata that would be important. And we utilized the RID initiative and that tagging system that was created by Anita Bondrowski and others to start unambiguously identifying all of the resources within HIRN. Now, we can access the dkNET API, and that makes it helpful for us when we want to look for certain resources and determine whether or not they have already been registered or identified. But we can also provide access to our API. So I wanted to show you what that looked like as well. Now, our API is provided to anyone who might be programmatically savvy. You want to grab some JSON, that's fine. And if you know what you're doing, you can do that. And these endpoints here, the P designation means these are public endpoints, and the R designation means that they're restrictive endpoints. But you can just get in contact with us and just get an API key, and you'll be able to access that data. We also provide information about some of the models that we use. But what if you don't know how to use the API at all, directly that is, then what do? So actually, Enhancement in my lab created a package called Sorcery. And this is an R package that was designed to interact with the API. And so all of the documentation on how to use that R package are online, and I'm not going to get into the details of that here, but I will give you a couple of examples of how you can do that. So one question that you might ask is, are there any problematic cell lines produced or used by HIRN? Maybe you're considering using a cell line, but you don't know if that cell line has ever been reported to have any problems. The idea is that the API can be used to integrate into some sort of monitoring service, maybe for cell lines or maybe for other resources, and just to check the quality of the inputs and outputs through HIRN. Now, another question you might ask is, does HIRN have any data sets similar to mine? So maybe you've got a particular kind of data set and you're trying to expand the utility of your data set by looking in HIRN and seeing what is available there. Now, these searches are done based on ontology and author attributes, and can give you some idea of, from a hierarchical point of view or a relational point of view, what other data sets exist that might be of interest to you. Now, there's a third thing as well that you can do, and you can contribute data in batch. So there's actually two ways you can interact with the resource browsers. You can directly report resources to the browser. Also, if you're not a part of HIRN and you've used some of these resources and you want to report either in a good way or a bad way, so meaning I had a bad experience with this antibody, it didn't work, or this data set was fantastic and I used it for another purpose, you can do that here as well too. Now, and so we've got some documentation to show you how to contribute your data in batch. So this is just kind of a brief overview of this first resource, and I just wanted to show you the team that is responsible for developing that resource and maintaining that resource. We get a lot of help from various individuals, but I will call out in particular Dave Coe and Enhancement from the lab, and they're really have been doing the lion's share of building the systems, documenting that, and helping to interact with the data. So next, we move to the PancDB, or the pancreas DB as it's better known. And this is a pretty large group within the HIRN network that I was telling you about. And the primary goal of this group is to accumulate, analyze, and distribute a lot of these high-value data sets to the diabetes community. And it's not only the type 1 diabetes community, but also resources for type 2 diabetes. So how is this done? This is a busy slide, but to the left, what you see is essentially the intake procedure. So these are actually people that die, and when they die, they donate their pancreas and other organs to this program. And these organs and these tissues are then taken into a lab and they're deeply characterized. So when I was saying deep phenotyping, what I meant is you can take the tissue, you can do histology on it, you can do genomics, you do physiological measurements, you can do some immune cell studies, and then all of that data gets into this pancreas database. And so where do you access this database? It's actually also freely available online. You can grab the data sets now. You're only asked to acknowledge that in your papers or abstracts, and there's a way that you're told to acknowledge the data, but it's freely available there. So I'm going to show you the kind of information you can get from this portal as well. This is kind of the opening screen, and on to the left, you can see that the database will enable you to search through the different individuals that have donated their pancreas and or other tissues to the program. And then what you can do is you can take these cards of information, and you can quickly kind of get an idea of what some of the underlying data looks like, whether you're looking at some genetic information, characterization of the different cell types, what types of antibodies the individual may or may not have had. Then you can also specifically pull and compare these data points across different individuals. So you might be interested in a subset of these features, and you can pull that data. Also, what you can do is you can start to look through individually in some more detail this information. And like I said, this can be readily downloaded. All of this data can be readily downloaded. And so then what you can do from there is when you actually want to download the data, you click on these different features that you want, and you can take that to your machine, put it on an HPC, a high-performance cluster, depending on what type of data that you're downloading for analysis. And then you can also actually go and get some imaging data. And you have this imaging data available through this pancreas database. But this actually brings us to our third portal, which is the Pancreas Atlas. And so what the Pancreas Atlas does is it essentially takes slices of the human pancreas and it catalogs that in such a way that you can analyze it in a bit more detail. And so this is an effort where you first need to prepare these metadata, and you need to take scripts and retrieve the images from their original storage locations. You process these images, and I won't get into the details of how you do that. And then you take the collections of images and you make those available to people. Now, the team that's been working on that is pictured here. This is efforts are being led by Marcela Brasova and others. And you can find that homepage. Again, it's publicly available for you to access here. And so what you do is once you get into the system, you can pick what tissue collection you're interested in exploring. And from there, you can start to filter your data. So you can filter it on disease status. You can filter it on the duration of the disease. So what gets tested is individuals from a very young age to individuals from individuals with long-standing disease. And so sadly, you have very young people that die and donate their pancreas to the program. And thankfully, we can learn a lot from these the donations that people make. And so you can look to get get any of these images, and what you see are things called H&E sectionings, and you can actually take a look at the whole entire pancreas that way. And then you can do some dynamic filtering based on what program you might be interested in exploring data from. So you can also filter by modality. So this is an easy way for you to access, for example, all the imaging mass cytometry data collections. And then what you see here colored in all these different colors are these different channels and these different markers that you can use to color these images of interest. So here is a pop-up example of what that might look like, where you're looking in at a particular islet section, and you can see all of the different markers that have been used to color this. And of course, you can strip this down to just a few of the images or a few of the markers that you want. And then you can also go back to the pancreas database that I was showing you, the second portal, and you can see what information is available there. So these different portals link to one another, so you can get different layers of information about these individuals that have donated their pancreas and other organs. And so now here what you have is a web browser that's highly interactive, and you can essentially get a thumbnail image, and then you can zoom in and out and navigate around using the top menu bar. And then on the left-hand side, what you see is a channel, and these different channel groups allow you to then select what you want to see. So if you want to look, for example, at the extracellular matrix, ECM, you have this purple color, so you can see that the extracellular matrix is all around the human islet. You can look at different immune markers as well, too. So general leukocyte markers colored in this kind of bluish type color. You can do that as well, too. So you can easily maneuver and toggle around there. And so here we've toggled on the immune channel, and so you can visualize an immune cell in some detail and in some action. But what if instead of just viewing the immune cell, you want to actually look at the sequencing data on the immune cell? Well, that brings me to my fourth data portal, and that's the clone search portal. And so here what is happening is we are taking collections of data, not just from the pancreas, but from other regions that are important in immunology, and then we sort these individual cells, and then these cells become sequenced. And then what we do is we take all of that data and we prepare that data, and then we serve it up as a web UI or a web interface. And so what you can then do is you can then take, either using an amino acid sequence or the nucleotide sequence, you could then search through something like 12 or 13 million different sequences, not only from the pancreas, but from peripheral blood and other regions, and then look to see if you can find any matches in your sequence of interest to other sequences of interest. And if you do find any matches, you can, of course, download all of that data and then, take that and use that for other purposes. So this is also a fourth tool that you can use. So in summary, I started by showing you type 1 diabetes and what it looks like. And what we've described here is a collection of different groups that are focusing on different aspects of type 1 diabetes. And these 4 different portals are capturing the data that's being generated from the top, and it's organizing it into different views, different ways of analyzing the data so that you could then access it. Now, there's something else I didn't talk about today, but I do think it's important to acknowledge, and that is the ongoing efforts that some of you on the phone will be aware of. And we keep these principles close by when we think about the data and how we can make that available to others. So, in terms of the spectrum, we slowly start to move to increased levels of fairness, but that hasn't always been the case. When we first started, the notion of fair had not been firmly established yet, and so we've been really using our collaboration with dkNET to help us and guide us in terms of how we can improve the unambiguous identification of resources, registering these resources, preserving them, and making them available to investigators. It looks like I've kind of quickly went through everything. I thought this would take a bit longer than it did. I do nothing. I just make slides and write papers. But this is a collection of a lot of different people doing a lot of different things. So these are the different labs, some of the different labs that were focused on the portal in particular. So from my lab, Annuet Vu and Dave Koh have been major contributors. Joyce Nyland is also a major PI of the HIREC grant, and a number of people from her group have also been instrumental in getting this data platform to where it is today. The last portal that I presented to you, the clone search portal, has been a collaboration with Todd Brusco's group from the University of Florida, and in particular, Howie Saya. Klaus Kausner kindly provided the slides for the pancreas DB work, and so this is being led by his group and others. The imaging catalog was prepared by Marcella Brisova, and she kindly allowed me to share that work with you today. But this is work that she and others are doing at Vanderbilt University. And of course, I can't overstate the contributions of people that actually donate their pancreas, and make those available to our communities so that we can study and learn more about this disease. The work is funded by a number of different agencies. We are funded by the NIDDK. We're also funded by JDRF. There's been a lot of overlap between the HIRN community and the MPOD community, which I did not speak a lot about today. But these two groups interact with each other. So I hope I've given you sort of an overview of the resources being made available through HIRN. I've collected the links that I presented during the presentation, and those are available for you here. All of the resources are available online, and you can access any of these. If you have problems, suggestions, questions, I am very keen on receiving any of these comments and feedback, and perhaps we may collaborate one day together. So I think with that, Koei, I'll turn it back over to you, and we can go from there.
Okay, thank you for John's presentation. So now, if you have questions, you could unmute. The unmute button is on the lower left side of your control. Let me also check. If you don't have a speaker, you can also use chat function. during the Q&A section. Let me check the chat. Okay, there is one question. I think it's for PancDB. Yeah.
Okay.
Yeah. So you mentioned about research can pull data and compare the data, the clinical data there. Is there
Yeah.
a function to let them compare? Or just they need to download and then compare by themselves?
So it depends on the kind of data that you are interested in. So for example, if you were interested in the imaging mass cytometry data, you wouldn't do that comparison directly in PancDB. You would use the pancreas atlas for that. Another example, if you had ATAC-seq data or RNA-seq data, you wouldn't directly do the comparison in PancDB. What you would do is you would download that data, and you would have to have some expertise to then take those raw data files and do the comparisons there. The comparisons that I was making reference to was comparisons of clinical data and some other descriptive types of data. Like, for example, when we were talking about the human islet and you were talking about the different cell types there, what the HPAP group has done is they've characterized the composition of these islets, so you can know how many beta cells are there or how many alpha cells are there. And so that data is available, and you can do comparisons directly on the site there. But for a majority of the data, the really large data sets, you can download those and then do comparisons and analysis using expertise that you have or that might be available to you at your institution.
Okay. Thank you. Let me check. There is one more question. I think it's from earlier. I assume it's for the resource browser. For the resource you mentioned, such as antibody, can we request it or we have to be a member?
No, you can absolutely request it. And what you should see is when you go there, you should see some key contacts. So for example, if you look at an antibody and you want to know how that antibody was used, so if it was a commercial antibody, you can be linked to the vendor for it. If it was an antibody made by members of this network, you'll have the contact information provided for you there, so you can directly contact those individuals. Likewise, if it was an antibody that was prepared by a vendor, but you want to contact someone that's actually used it, so you want to know beforehand, hey, was this really any good? We've made the contact information available there for you, so maybe you want to ask and get a bit more candid advice or opinions about those antibodies. You can certainly do that there, and that information is provided.
Are there any more questions? I don't see question from chat box. So if you have any more question, please feel free to send us an email. I will forward it to Dr. Kaddis. So I think thanks for John for the great presentation today, and thanks for everyone joining today's webinar. I hope you enjoyed today's webinar. A recorded version of this webinar will be available on dkNET YouTube channel next week. We'd like to encourage you to share today's webinar with your colleagues and students. Our next webinar will be on April 24th, and we will introduce you another useful resource, AppGene. There is another announcement from the dkNET. Let me share the screen first. Okay. So the 2020 dkNET Summer of Data Student Internship Program is accepting applications. The internship is designed to provide students an opportunity to use the dkNET information portal and resource for their research project. Students will learn the best practice to enhance rigor and reproducibility and learn the basic of good data management by following the fair data principles. This is an online program, so the duration is 6 weeks. Students will attend a 1-week online bootcamp, 2 hours per day, and then for the last 5 weeks, they will attend the meeting once a week to discuss about their project. So if you have or you know any student that will work in a research lab during this summer, please encourage them to apply. $1,000 scholarship will be awarded to a limited number of students. The application deadline is April 17, 2020. So if you are interested in learning more about this program or learning what our alumni said about the program, please go to dkNET.org and click the Summer Student Internship Program. Okay, that's today's announcement. To receive more information such as funding opportunity or upcoming webinar, please subscribe to our newsletter or check the announcement at dkNET.org. Thank you all again and have a good weekend. Thank you.